Hi everyone, welcome to this video on translation. So the process of translation is the final step of protein synthesis, and in this step we are going to be using the messenger RNA copy or the messenger RNA transcript that we built in transcription, and we're going to use it to build a protein or build a polypeptide strand that can then be folded up into a protein. So similar to transcription, there are going to be a couple main steps that are involved with translation, and this slide is an overview of those steps. So there are going to be a lot of words here that I haven't described or I haven't explained yet, but we're going to go through each of these steps one by one. But just for now, as an overview, we're putting them all together on this one slide. So the first step is that the mRNA transcript, the ribosome, and a tRNA molecule will have to come together at the start codon. Then the ribosome is going to move along that mRNA transcript, and it's going to be adding amino acids to the growing polypeptide chain. And in this image, the ribosome is shown here in this like pinkish color. The tRNA molecules are these sort of, I'm not sure what the shape is, like a T almost, T-shaped molecules here. And the growing polypeptide chain are these rectangles that are coming out. And then the final step is that we're going to finish at a stop codon, and the polypeptide will be released, so it'll come off of the ribosome. So let's go into a little bit more detail about how this is going to happen. So translation is going to happen at a ribosome. Ribosome is going to be kind of the workbench where we're going to build our polypeptide strand that can then fold up into a protein. So here we're showing the mRNA strand, and the ribosome has two sort of subunits. So it has a top subunit and a bottom subunit, also called a small subunit and a large subunit. So how the question is then going to be how the ribosome is going to translate, because we're doing translation, this mRNA transcript into amino acids. We know that the language of RNA uses 4 different bases. There's A's, U's, C's, and G's. But we also know that when we're thinking about the language of amino acids, there's about 20 different ones. So if we're translating between the language of RNA into the language of amino acid, how does that translation work when we're going from just 4 different kinds of bases in mRNA to 20 different kinds of amino acids? So when we're reading the mRNA, we actually group them into sets of 3. So instead of reading each letter in mRNA one at a time, instead of saying, oh, here's a, here's a U, here's a G, here's another G, here's another C, and a C, we're going to group the mRNA letters into sets of 3. So just like you read English by having specific letters grouped into words, you don't just read one string of letters that are all jumbled together. The words are separated. So B goes with Y, and then there's a space, and then there's GROU. So the letters are separated out into words. The words for mRNA are groups of 3 letters. So this long stretch of RNA bases is going to turn into these 5 different distinct little subsets. So A is grouped with U and G, and that's one set. GCC is another set. UAC is a third set. GGC is the 4th set, and UGA is the 5th set. So we're going to have 5 different sets or 5 different words instead of just this one long chain of letters. So each of these sets, each of these little words is called a codon. So one codon is a set of 3 RNA bases, 3 RNA nucleotides. So here are 5 codons right here. Codons are going to allow us to translate the language of nucleotides or nucleic acid to the language of amino acids. And to do that, we're going to be using one of 2 tools, or you could use both if you like. Here we have a codon wheel, and here we have a codon chart. But both of these will allow you to look at the genetic code. So the genetic code refers to what amino acids correspond to each particular kind of codon. So we can practice with a couple examples here. So here we have our 5 codons from earlier, and you can use either the codon wheel or the codon chart to figure out what amino acid corresponds to each of these codons. So we'll do a few examples. So AUG is our first codon. And if you look at the codon wheel, you're going to start on the inside. So you're going to start in this inner wheel here. And the first letter in our first codon is A. So we're going to look at this bottom left quadrant where it's labeled A here. And our second letter is U. So we're going to go from the A, and then we're going to find the U on the next outer wheel. So go A and then U. And then our final letter is G. So we're going to go out even further and look for the G in this final outer wheel, and we find it right here. So that tells us that our AUG codon corresponds to the amino acid methionine, which is abbreviated by MET or the letter M. And you can see the exact same information from this codon chart. So for the codon chart, you're going to start on the left-hand side and find your first letter. So that's A right here. So that's telling us that we're somewhere in this row, in this A row. And then we look for our second letter across the top. So our second letter is U, so that's right here. So if we look at the AU sort of cross section, we're going to be in this box right here. And then you look for your third letter either along this left-hand side, G is right here, so you can go all the way back here, or you can just look for your actual codon along the side here. So AUG corresponds to methionine. You'll notice that this says start underneath, and this has a little triangle that says start. So one characteristic of the process of translation is that a start codon is going to be methionine for pretty much every polypeptide strand. And if you go back to this first slide, it said the mRNA, the ribosome, and the tRNA are going to come together at a start codon. So now we know a codon is a set of 3, and specifically a start codon is going to be a set of 3 that corresponds to methionine. So AUG is going to be the start for pretty much every polypeptide strand, and then it'll be different after that start codon. So let's practice with a couple others. So we'll do 2 with the wheel and then 2 with the chart. So GCC, we're going to start in the upper left, and then we have a C right here. And then if you find the C outside here, you'll know that you're going to have an alanine for our second amino acid. And then UAC, we're going to start in the upper right with this green U, and then an A is next. So we're going to go out to this pink colored A, and then C is our final letter. So C is right here, and that tells us that tyrosine, or TYR, for some reason the one letter abbreviation is Y, tyrosine is our third amino acid that corresponds to UAC. And then I'll use the chart for the last 2. GGC is our 4th amino acid, or fourth codon. So G is down here in green at the bottom. And then G is our second letter in our codon. So we're going to go all the way down here across to the end where G is at the top. So G, this row, G, this column, and then C is our final letter. So GGC corresponds to glycine. And then UGA is our final. So here, U is where we're going to start in this row, and then we'll find G across the top, and then we'll look for A across the right-hand side. So UGA, and you'll notice here that it doesn't give you an abbreviation for an amino acid. Instead, it just says stop. And you'll notice that if you look for the UGA on the codon wheel, UGA, you don't get an abbreviation here either. You get a little square, and the square is going to correspond to stop. So when you reach a stop codon, the process of transcription is over, and that's going to cause the polypeptide strand that we've built to just come apart from the ribosome, and the process will be finished. So these stop codons, UAA, UAG, and UGA, terminate the process of translation. Okay, so when we're thinking about this process actually happening inside of your cells, the ribosomes that are going along this mRNA transcript don't have little codon wheels or codon charts as they're moving along. So how does the ribosome, which is the workbench for protein synthesis, how does it know what amino acids need to be coming in? So maybe this is UAG and we need a methionine. How does that actually happen? Here, the process is going to introduce these tRNA molecules that I mentioned on the first slide. So tRNA molecules are called transfer RNA molecules. So it's another kind of RNA. We had messenger RNA that's copying the information from the nucleus in the DNA. tRNA molecules are responsible for bringing around amino acids. So they're basically dedicated amino acid carriers that bring amino acids to the ribosome, and they allow the proper amino acids to be brought in that are going to correspond to each of the codons on the RNA. So how does this work? tRNA molecules match specific codons to their corresponding amino acids, and they're going to have a couple things on them. So I said they carry amino acids. So one end is going to be carrying around amino acid. There's different kinds of tRNAs for the different amino acids. So this tRNA is carrying methionine. There will be another tRNA that's dedicated to carrying alanine, one for tyrosine. So each amino acid has dedicated tRNA molecules that are going to carry that amino acid around. On the other side of the tRNA is what's called an anticodon. So the anticodon is going to be an RNA nucleotide triplet that's complementary to the codon on the mRNA. So here our mRNA codon says AUG, and we know that A is complementary to U for RNA, U is complementary to A, and G is complementary to C. So this anticodon is going to sort of like be attracted to, it's going to hydrogen bond with this codon on the mRNA. They are a match for each other. And that's how we match this codon, AUG, to the amino acid, because UAC is on the tRNA and it's carrying methionine. So the amino acid methionine is going to be matched with the codon AUG because of this anticodon being able to sort of sit down on top of it. So let's look at a picture of this happening. So we know AUG brings methionine, and we can see that right here. So AUG goes with methionine. So this is going to be happening in the ribosome, and here's our AUG. Here's our anticodon on the tRNA. It's carrying methionine. And then we have, we're just going to keep going along and reading the mRNA triplet by triplet, codon by codon. So our next codon here is UGG, and that's going to be complementary to this anticodon right here. So U matches with A, G matches with C, and G matches with C. So this tRNA with the ACC anticodon is going to sit down next, and it's carrying this TRP, tryptophan amino acid. So that's going to come in next based on this UGG codon. So translation is going to start with MET, codon AUG, and then the second amino acid is going to be added, and that's tryptophan here. So here we've got our first amino acid, methionine, and then our second amino acid got added here, tryptophan. So our next codon is going to be UUC. So we know that on the anticodon for the next amino acid, we should have an A, because that's complementary with the U, another A, and then a G. So the next anticodon should read AAG. And we can also predict if AAG is going to be the next anticodon, what amino acid it'll be carrying using the codon wheel or the codon chart. So our mRNA codon is UUC. So if we go U, then C, then we can see that phenylalanine is going to be the amino acid that we should be bringing in. So remember, when you're using the codon wheel or the codon chart, you're always going to be using it based on the mRNA, not the tRNA. So UUC is what we look up, not AAG. But AAG will be on the bottom of the tRNA. So here we see that coming in. AAG is on the bottom to be complementary to our mRNA, and it's carrying phenylalanine, our third amino acid. So like I said, there are 3 stop codons here. So if you run into a stop codon, the whole process is just going to finish. And instead of adding an amino acid, it's going to terminate the process. And our polypeptide strand is going to just sort of float away. So here, UAA is a stop codon, and instead of bringing in an amino acid, we're just going to finish the process. You'll never have an amino acid stretch that's just 3 amino acids long. It'll be hundreds or thousands of in length, but just for the process simplicity, we're demonstrating this concept, we're just going to say the stop codon happens here, and then we're finished. So that's the process of translation, and I will see you all soon.